Very warm welcome everybody. Come on. So let us begin our understanding today, right? So today we are going to have a look at your unit 13A, that is on your isolation of DNA. Rather, it is often looked as a very simple topic, right? But definitely it is going to be a scoreable one. Yes, Maya. So why I tell it as a scoreable one? Because you are getting questions like the problems that are related to the OD. They will be giving you up a OD and they will ask you what is the concentration of DNA. Isn't it? So these sort of questions are always feasible. Yes. So definitely never ever take any topic to be light on your CSIR. Yes. I hope that you understand this importance of this topic. Isn't it? So always don't have a bias thinking that isolation is a so simple topic. Why should I study? Definitely even in that questions are coming. Alright, yes everyone, very good evening. Come on. So probably you might be knowing me. I am Dr. Saranya and already have done my PhD in biomaterials, and I do have 13 papers of good impact, and obviously I have already cracked NET as well as the SET examination of our state. And I do have 10 years of teaching and research experience. I have been associated with the top notches of your CSIR NET coaching institutes, like I have been with Biotechnica and all. So now I am teaching live on your Unacademy Plus. Yes, so far greater than 100 plus students have already cracked from my batch. Right? Fine. So let us see in detail what we are going to do it up. Yes, so if at all you are new to this CSIR NET and you want to know what you really want from it. Yes, so you have two options. One is to aim for a JRF or else for your LS. So if you have the age eligibility, probably you can take up for the JRF. The cutoff is quite high relatively, but if you crack it, you will be awarded a stipend of almost 40 plus, right? So with this, definitely a PhD you can reach up with the stipend. Right, so that is the most prestigious thing for everybody like us, right? So you can become a scientist with a pay. All right, and also keep reminded, if you are telling somebody that you have already cracked NET, that's it. So your entry is becoming very easy anywhere. Let it be an industry. I have been with industry also, so I know that if at all a resume is coming, the first one we will check out is that JRF, yes, or at least LS. Right, so if it is there, it is a green signal for your resume. All right, so this is what definitely it is your need. So take it seriously. Time it is little bit extended for you. Your exam is only in July now, approximately. So make use of the time and learn a lot. Definitely you will crack the examination. All right, so now coming to this class, it is on your isolation of DNA. All right, why do you want to isolate a DNA from the cell? What is the need? Yes, why really you want to isolate the DNA? Why not? Yes, why it can be, why it needed to be isolated? What is the importance of it? Yeah? So whenever you are isolating, then only you will know everything about it. For example, you are interested to make a sequence of it. Yes, fine. So if you want to make a sequence, obviously what you must do? You should isolate your DNA. Without isolation, how will you know about the sequence? You cannot know it. Yes, and then if at all you want to develop a synthesis product, you want to clone it. Before that, you should isolate your DNA. Then only you will be able to make all other process. You want to do a product kit development. Yes. Now see, you are using your all your PCR techniques. You want to convert it into a transcript. Definitely for all those, your isolation is a very crucial step. So don't miss out this isolation part. All right. So next, what you can do with this optimizing your isolation. Why your isolation is thought of as so much important step everywhere. Yes, this isolation, it is typically helping you to maximize your DNA recovery. Maybe you are working with a crime site, right? So you have the victim's blood, right? So you get only 1 ml of the blood, right? So what you should do with that blood? You should absolutely, yeah, you should maximize the recovery out of the sample. Yes, then only you will be able to carry over with the next step, like you will be probably amplifying them by using your PCR, right? Yes, and then you have to probably remove all the inhibitors, or else your isolation step becomes very tough. Yes, what about these nucleases? What do they do with us? Yes, What will a nuclease do? Yes, everyone, come on. So what will the nucleases do? They are going to degrade your DNA. Right. So if they are degrading your DNA, you have to remove them. Isn't it? So how you will remove them? Yes, you are going to treat them. Right. You are going to inhibit the nucleases. Then it is easy for you to maximize your recovery. Right. So all these steps, it's what we are going to see today. How we are going to isolate your DNA in a proper manner. Absolutely, Pushpanjali. So what it does, it is breaking up your DNA. Right. So if at all you want to isolate your DNA, what you should do? Yes, you have to first break open your cell. Right. So there are many methods to break open your cell. Right. So the first method, it is using your chemicals. Right. So what are the common chemicals that you use to break over a cell? You can use the SDS. What is SDS? Yes, it is a detergent. What sort of detergent is SDS? What sort of detergent? Yes, Supratim Ghosh. It is going to be an anionic detergent. What do you mean by anionic? It is a negatively charged one. Absolutely. If you take the CTAB, it is going to be a cationic detergent. Yes. What do you mean by cationic? It is a positively charged detergents. Got my point? Right. So these are the chemical methods by which you are lysing the cell. Right. Also, there is going to be the enzymatic method of making a cell lysis, that you can definitely use your proteinase K. Yeah. So that is a good enzyme, but it is quite costly. So in industry and research, we don't use them much because it is very costly for you to afford. Rather, you can use other normal methods. Apart from that, you can use the mechanical methods. Absolutely. So all these mechanical methods like your grinding, freeze drying, all those things will help you to lyse your cells. Right. So what is present inside a cell? Come on, quickly, very quickly. You take up a cell is made up of what? Yes. So will it contain the protein? Yes, absolutely. Yes, Meera, come on. Yes, Meera. Right. So it will contain the protein, right? And then you have your nucleic acid. All right. You have a lipid. Yes. So you have lipid also. So protein, you are removing them by all these digestion process. You have to remove the lipid also. Yes, then only you will arrive at your nucleic acid. Right. Isn't it? Right. So how you will remove your protein now? You are going to use, absolutely Maya. You have your carbohydrate, you have your lipid. So you have to remove everything. You want only your nucleic acid. You want only your DNA. Right? And so now you have broken only your cell wall or your cell membrane you have broken. Now all the contents are present inside alongside with your DNA. You are going to remove it. So how you are going to achieve it? You can achieve it either by using your organic solvent. What is an organic solvent? Yes. What is an organic solvent? The best example is going to be your phenol. Right. So what is the mechanism of action of phenol? So phenol, what it does? This phenol, yes, it is going to react with your molecule. Absolutely. Yes, very good evening, Satya Maithili. Right. So what your phenol is doing? See here. Your DNA is going to be a negatively charged molecule. Yes, absolutely. Yes. So it is going to flip your protein. I'll tell you how. Your DNA, it is going to be a negatively charged molecule. So it is a polar thing. Am I right? You agree DNA is polar? Right. So you have heard that like will dissolve like. Yes? True or false? Like will dissolve the like. Absolutely. So your DNA is polar. So it will go only with the polar solvent. Yes. So what about your phenol? Phenol is, what kind of material is phenol? Come on. So phenol is polar or non-polar? Yes. What is phenol? What kind of material is phenol? See here, phenol is going to be less polar. Remember that. Always. Your phenol, it is going to be less polar. Never. Phenol is less polar. Only your water molecule, it is going to be polar. Alright. Yes. See here. Your water molecule is only polar. Your phenol, it is going to be less polar relative to water. Yes, I agree that OH is there, but still, yes, relative to water, it is being less polar, right? So now what happens? I told you that like will like only same, isn't it? Fine. So protein, they have your polar as well as your non-polar residue, right? So phenol is becoming less polar when compared to your aqueous solution. Yes, what is an aqueous solution? Aqueous solution will contain more of water. You got it? Yes. So what you are doing, you are taking your cell extract, right? So see here, your cell extract is there. To that, you are adding the phenol, right? So cell extract, hey, water is polar, no doubt. Yes, yes, water is a polar solvent, no doubt in that. Yes, absolutely. Water is a true polar, yes, Shrisha. Come on, yes, Shakshi, water is a polar solvent. Yes, your phenol, it is relatively, it is a non-polar solvent. That is why you are getting two distinct layers. You got it? Yes, Sakshi, you understood it now? So now what you are doing, you are mixing your cells along with the phenol, right? And then you are making a spin. So once you spin them, what happens? It is separated into two layers, right? Yes. So the top layer, it is becoming your aqueous layer. Aqueous means it is a water rich. Oh, thank you, Smiley. Yes, yes. So this is going to become your water rich aqueous layer, right? And then in the interface, there is going to be your protein. Alright, so all the coagulated protein, it will be in the interface, and in the bottom phase, there is going to be your phenol. Your phenol is non-polar, and all the major proteins, they come here in the phenol region. Yes, so this coagulated protein will be in the debris. Got it? Absolutely. Yes, Shweta, absolutely water is polar solvent. No doubt in that. Yes, your water will be on the top, your intermediate or your coagulated protein will be in the middle, and all your dissolved protein will go down. This is the basic principle behind the separation of your nucleic acid by using this method of your organic solvent. Right? So first you understand this. Now we will see how this phenol is helping in the separation. Right? So you are adding your cells. In the cell, DNA is there. Yes. Proteins are there. Absolutely. You got it? Right? Yes, yes. So both your DNA and protein are there. Now you want to separate the protein from your DNA. How you are separating it? See here, your DNA is going to be charged. It is a negatively charged protein, right? So this becomes polar. So if it is polar, where it will go? Yes, you have two layers. One is the aqueous layer, other one it is your non-polar layer. So your DNA will go to the top or bottom. You understood my question? You understood my question? See here, you have a layer. All right. Yes. See. So here water is present. So this is a polar one. Right. So this is going to be a non-polar one. Yeah. So your DNA, it is negatively charged. Absolutely. You got it now? Yes, Supratim. Yes, Sakshi, Shrisha, Mayank. Absolutely. Since because your DNA is charged, right? It is going to the top phase. It is going to the aqueous phase. Got it? Yes? Absolutely. Yeah. Yes, Aradhya. So your DNA is moving to the top phase or the aqueous phase. Right. So what happens to all your proteins? Yeah. So they are moving to the bottom phase or the phenolic phase. Right. So now a natural doubt should come to you. Aren't my protein polar? Aren't my protein charged? Yes, the doubt came or what? I agree that DNA is charged, so it is going to the top. Yes, so you agree with that. What about my proteins? Why should my protein go to the non-polar region? My proteins are also charged. Yes, right, your doubt is valid. But what your phenol does? See, in general, how will be your protein? Yes, in general, in your protein, the water fearing region will be inside. Okay, so all the hydrophobic will be inside, right? And your hydrophilic will be outside. You agree this? Yes, all the hydrophilic region will be outside because it is going to interact with the water in general. Yes, right. And inside, how it will be looking like? Inside it will be hydrophobic. You understood this first? Yes, this is in general. Okay, fine. Now you are adding the phenol. The moment you add the phenol, this protein, it is going to flip. What do you mean by flip? This hydrophilic is going inside and your hydrophobic is coming outside. You can see it here. So with your less polar residue, they are flipping outside. Initially, blue was outside, green is inside. Now it is flipping. So green is becoming out. So green is going to be phobic. Right? So if it is hydrophobic, it will be always interacting with your less polar or non-polar solvent like phenol. Right? So that is why once your phenol is added, the protein is flipping. Yes. So even now your lipids also, they follow the same principle. So they become soluble in the phenol, or you call them as your non-polar phase, right? And your yes, so your water, not water, I'm just telling you, all your aqueous phase. So how this aqueous phase has come? Yes, aqueous phase is rich in water. Yes, so how this aqueous phase has come? It has come only from the cell. You didn't add water anywhere. Yes, your aqueous phase is going to be all your polar residues, right? So all the polar content along with your DNA. So that is going to be in the aqueous phase and your protein is going to your non-polar phase. So now you have separated these two regions carefully. Got the principle behind this? Now you will always understand why you are using your phenol chloroform in this technique, right? Fine. So that is how you are removing the protein by using your organic solvents. Phenol will always help in the removal, right? And or your destruction. And chloroform will keep them in solution. That's it. So that is the duty of the chloroform. Alright. Like that you can use your salt. You can use your salt for this. You can use your DNA binding. Yes, Meera. So that's what. The same thing if you take a lipid. So what happened to the lipid? Even lipids, they are going to be your hydrophobic structures. Right? So even they are kept under, like your protein, they are kept under your phenol residues. Right? Yes. Carbohydrates. What happens? When you treat the carbohydrate with phenol chloroform, they are very aggressive. They are very corrosive. So they are being degraded into chalks. Right? So that is why even they get into the debris and they have high probability to settle up in this thing. Right? Fine. So next you can use your DNA binding beads. Yes, you have your Kelex beads. So what they do? All the protein, DNA, everything others, so protein, lipid, everything will be left in the solution. Only your DNA will bind to the bead. The bead will probably containing your guanidinium thiocyanate. So it will have a rich positive charge. So your DNA is negative charge, it will go and bind. So all others will remain in the solution. So likewise also you can separate them. Alright, finally the final step is you will be adding an alcohol. Probably you will use an ice cold alcohol, yes, such that you will get a sort of stability into it. Alright, and then this is the total procedure. Now we will see some more inner details. What happens if you use this SDS? Yes. So SDS, it is going to be a detergent as I told you. So this becomes incorporated between your plasma membrane. Right? Fine. So now what happens? So this SDS, it is containing a hydrophobic tail and a hydrophilic head. isn't it? So they get forming your mixed micelles, and so they are going to lyse your cells, right? Absolutely. So your cell become porous. So lipid, they are removed not only by your this thing phenol, but also your detergents. They are very efficient in removing the lipids. Yes, Meera, I think your doubt is done now, right? Fine. So apart from this phenol is to chloroform, your organic extraction method, you can use your non-organic method, you can use your nylon membrane method as well, right? So this is how you do up with your organic extraction. Yes, so you take your lysate. So this lysate will contain your nucleic acid, and now you are going to mix them with equal volume of your organic solvent. Yes, and then you centrifuge them. You have your aqueous phase. So the aqueous phase is going to contain what? The aqueous phase will contain yes, everyone. So the aqueous phase is going to contain your DNA. Yes, interphase is going to probably contain the debris, right? The organic phase is going to contain your protein. So you remember the protein flipping also. So you collect the organic, you collect the aqueous phase. Absolutely, you will have your DNA. All right. Yes, yes. So this is how you are going to isolate your DNA. Clear? Yes. Fine. Yes. So apart from that, you can use your lysozyme. Yes, if at all you are going with a gram-positive bacteria. So this will digest your peptidoglycan layer. All right. Yes. So for your plant, you can use the cellulose, because plant cell wall is very, very thick, right? And for your plants, you don't do SDS. You use the CTAB method. We'll be very shortly seeing that. And with the yeast, you do with your zymolase. All right. So I told you that plant DNA extraction, you use a CTAB method. What is this CTAB? CTAB, it is going to be a cationic detergent like SDS. This CTAB, it is a cationic detergent. So what it will do? Our DNA is going to be negatively charged, and this CTAB, it is going to be positively charged, right? So they both have a very good binding with each other, right? So now if you add the protein and nucleic acid, your CTAB is going to bind with the nucleic acid, right? So it is going to go to the pellet. Then you resuspend them in your NaCl, you are getting the stuff, right? You can precipitate them using ethanol, so you are getting the pure DNA, right? So shall we compare? In phenol is to chloroform, where will be the DNA? Yes, phenol is to chloroform, the DNA is at the top, right? So in case of your CTAB, where is the DNA? It is in the pellet. You got the difference between these two? Yes, right. Because here your DNA is excluded. It is going to your aqueous layer. But here your detergent itself is going to be absolutely binding with it. So thereby it is in the pellet. Yes, everyone. Absolutely, you are correct. Right. So let us take up a question. All right. Yes. So which most common step while extracting the DNA? Yes, it is going to be the use of organic solvent like your phenol is to chloroform. So what is the significance of phenol? Right. So an addition of phenol, your DNA is dissolved in the phenol. On addition of the phenol, your protein flips and it becomes soluble in phenol. DNA precipitates in the aqueous phase because of flipping, or all the above. Which is going to be the correct answer? Yes, very good. Yes, Soham. Yes, Shrisha. Absolutely. So if you add the phenol, just now we saw that your protein is flipping, so it is becoming soluble, because you know that your protein, it has both your hydrophilic and the phobic region. Your phenol is non-polar, so definitely it has to flip. If it is not flipping, it is never going to dissolve. Yes, Labdi. Absolutely correct. Right? Fine. So next question. A purified virus, yes, actually, you have purified a virus that is affecting your turnip leaves. Got it? Right. So you are treating them, a sample, with the phenol, right? And it is going to remove your proteins. All right, fine. So now you want to find what sort of material it is, whether it is a DNA or RNA. Right, so you don't know, you have just treated and kept your DNA isolated, you have kept the thing isolated, nucleic acid isolated. You don't know whether it is a DNA or RNA. So which method you will use to find? You will treat one with phenol-ester chloroform and other with DEPC. What is this DEPC? DEPC, it is your diethyl pyrocarbonate. Okay, so it is serving as an RNase inhibitor. Okay, right, but that is not under this review, but I am just helping you to understand that. Right, so it is to check for your infectivity, or you treat the one with ice cold ethanol and the other with your ice cold isobutanol and check for infectivity, or absolutely, yes Pradeep. Also you are going to treat the one with your DNAs and you are going to treat the other with your RNAs. Yes, and then you are finding the infectivity. Absolutely. Yes, Labdi, Sada, Supradim. Yes, Soham. Absolutely, you are correct. So option C is good, isn't it? Yes, yes, absolutely. Your option C is exactly matching our need. You want to find whether it is a DNA or an RNA. So if it is going to be a DNA, yes, it will be degraded by DNAs. So there is no activity. Got it? If it is going to be an RNA, you add the RNAs. So there is going to be no activity. Got it? Right. So definitely. So you might get a such of things. Yes. Right. Oh my God. Start codon. Nice, nice. Come on. So by using your CTAB for purifying your plant DNA, yes, which region will contain your DNA? Yes. So in your CTAB method, which region is going to contain your DNA? Whether it is going to be your supernatant, whether it is going to be your pellet, whether it is going to be both an interphasic. So this is A, this is B, this is C, and this is D. Absolutely. Your option B, that is your pellet is going to contain your nucleic acid. No doubt, your pellet is going to contain your nucleic acid. We told that CTAB is positively charged, so it is going to bind with your negatively charged DNA in the pellet. Right? So very well, you have isolated your DNA. Now you have to check them for your purity. Yes, 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 Raksha. Welcome to the class. Thank you for your wishes. Alright. So I told you that once you have isolated the DNA, yeah, you have to check. Absolutely, Anuja. Yes, Aradhya. So you have to check for your DNA. How you will check? You simply run them in a gel. If you are getting a streak like this, what do you infer from that? Yes, Aftab. If you are getting a bulk thing like at the end, it means that it is all your debris. Maybe it is an RNA contamination, or else your DNA is not digested properly. It got digested properly. It has come as a streak. So it is not a good quality. So when it will be of good quality, when it is a distinct band, it is called as a good quality band. Got it? Right. So this fourth lane is a good quality DNA. So all others, they are degraded because of the improper technique. Right. So next, how you are going to analyze your DNA? Yes, absolutely. So in general, the nucleic acids, they have the feature of absorbing your UV light. Okay. So how do they absorb the UV light? Yes, it is because they have the sugar phosphate backbone. Yes. So all this nucleic acid, they have your carbohydrate, that is the sugar, your ribose and all the stuff, deoxyribose. So along with that phosphate group. So both your DNA and RNA, they are capable of absorbing your UV. Yes. That absorption you are measuring. Right. So if you compare, you have your double stranded DNA, you have your single stranded DNA. Yes. So now who is giving a more absorption? Your single stranded DNA, it is showing a more absorption. Yes. So what happens to your double stranded DNA? It is showing less absorption. Yes, absolutely. At 260, if you check, yes, this is for your single stranded. It is showing more value. This is going to be a double stranded. It is showing a less value. Why it is showing a less value? It is showing a less value. Why? Because see, this double stranded DNA has a problem of stacking up of bases, right? See, if you are free, you can express anything, right? If some kind of one more fellow, like maybe your parent, yes, so if they are there, you may not express yourself properly, yes, right? So this is exposed more, so the base is more exposed. When the base becomes exposed more to your UV, this absorption is going to increase. Here it is going to be less exposed because it is being stacked. Somebody is hiding. So the base is, your backbone, it is being suppressed. So there is a less absorption. Yes, this is what is called as your hyperchromicity. Yes, it is increased one. This becomes your hypochromicity. Hypo means less, hyper means increased. Got my point? Right. So now you are measuring the OD at 260 nanometer. Alright. So if you take an one OD, right? So if at all the material you are containing, it is going to be your double standard DNA. Yes, it is approximated that one OD is going to correspond to 50 microgram per ml of your DNA. Yes. So if it is going to be 2 OD, so how much is my concentration of my double stranded DNA? Yes, it is going to be my 100 microgram per ml. Got it? So if I getting 3 OD, that becomes 150 microgram per ml. If my value is going to be 4 OD, yes, obviously, right? So you got my point. Good. If it is going to be a single stranded DNA, you have to multiply them by 33 microgram. Got it? Yes, because they are absorbing more, right? So only less people, right? So 33 microgram it is going to be for your single stranded DNA, right? Your single stranded RNA is going to be 40, right? So shall we just sum it up? If it is going to be a single strand, if it is going to be your double stranded DNA, it is going to be 50. If it is going to be your single stranded DNA, it is going to be 33. If it is going to be a single stranded RNA, it is going to be 40. 50, 33, 40. Got it? Clear? Right? Alright. Fine everyone. Right. So now let us take a problem on this. So these kind of problems are always coming up in GATE. Right. So there is a high time probability that even I think a couple of times it came in CSIR also. So there is a high time probability that these questions will come. So definitely you should learn to solve these questions. See this question. This optical density of 400 base pair long 1 ml DNA solution, it is going to be 0.052. What is it? 0.052. So how many DNA molecules are present in the solution in terms of your base pairs? Right. So one base pair is equal to 650 Dalton. Right. So the optical density of one OD, they have given it already. Yes, it is going to be around 50 microgram of DNA per ml. Right? Fine. So your one OD, it is going to be equal to 50 microgram of your DNA per ml. Right? Fine. So what is going to be the total DNA molecule that is present in the solution? Yes? Time is less. I'll just solve it for you if you don't mind. Yeah? Alright. I'll just directly give the solution. I don't have time. Right. See here. Your one OD is corresponding to 50 microgram of your double stranded DNA. Isn't it? In the question, how much OD is given? It is going to be 0.05 DOD. Right? So what you have to do? You have to multiply this 50 into this 0.052. Right? So it is getting around 2.6 mu g per ml. That is what we did, isn't it? So if it is going to be 2 OD, I told you that it will be 100. So this is going to be 0.052 OD. So that is going to become your 2.6 microgram per ml. Got it? Right. So the average weight, yes, of your one nucleotide base pair, it is 650 Dalton. Yes, right. So now if it is going to be a 400 base pair, right? So see here, they have given that it is going to be a 400 base pair. Yes, so which is going to, your one base pair is going to be 650. Now you are having 400 base pair. So what you have to do? You have to multiply this 400 by this 650. Got it? Yes. So now you are getting this many Dalton. So this is the average weight of your one nucleotide. Right. So now how to find with your number of moles? Yes, your number of moles is very simple. It is going to be your, it is going to be the ratio. Yes, it is the given weight by your molecular weight. Right. So given weight. Yes, so it is going to be your 2.6 into 10 to the power minus 6, right, divided by the molecular weight is going to be your 2,60,000. Absolutely. So this becomes your given weight, this becomes your molecular weight, right? So if you are going to divide it, you are going to get your 6 point, you are going to get your 1 into 10 to the power minus 11. Yes, this is going to be the number of moles, right? So now you have, for 1 mole you have 6.023 into 10 power 23. What is this? Your Avogadro number. Now you have 1 into 10 to the power minus 11 moles. Right? So it is going to be your 6.023 into this fellow. Right? So this becomes 6.023 into 10 to the power 12. So your answer 1 is going to be correct. Got it, everyone? I hope that this calculation is now understandable. First you have to understand how to calculate your given weight, how to calculate your molecular weight, and then what is the number of moles, and your 1 mole is going to contain this many molecules. So you will be arriving at the solution easily. Got it? Clear? Alright, everyone. Fine. So then you know that your purity, if you take the purity, it is going to be your absorbance at 260 by 280. So this is telling about the purity. Alright. So I have a couple of questions. You have lack of time. Yeah, I'm just moving in. Right. So you can quantify your DNA using your fluorescent dye method also. You can use your Hochet. You can use your ETBR. You can use the Pico Green. Right. So these 3 dyes are used for quantifying them by using the fluorescent method. Right. Okay. Only green you are not using. Only ethidium bromide, Hochet and Pico Green you use for your quantification. Alright, so that's all the information about that. And definitely at Unacademy we are giving it a quality education. Yes, and what you will get by this courses. You will get the access to all the 13 units. Yes, all the 13 units are getting covered. You will have a separate faculty for part A. Totally we are a team of 15 plus educators only for your life science. Right? Yes, so you will have every syllabus to be covered. So don't worry about the syllabus completion. The total units are getting covered. Oh yes, Mayank, thank you. And you will get a lot of practice MCQ with detailed solutions, like how we are taking up a question, explanation, like that. We do have a revision package. Everything is included in this. We do have your unit test, week test, mock test, doubt clearance sessions. And another feature, oh yes, Smiley. So another feature is there, it is called as poll, right? So all the package you are getting a benefit at 10,500. Yes, absolutely. So this is going to be a very less price for this course. I believe that you will be understanding how much cost effective it is going to be. Apart from that, you are going to get a 10% off on the total fee. You can use my code SaranyaLive. Right? Okay, so this is really going to be a wonderful package. And this month I have started with molecular interaction. We are dealing it in a simplified manner. Yes, and then I'm going to start with fundamental and developmental biology. Oh yes, yeah, thanks a lot everybody. And parallelly I am running a PYQ session. So I strongly recommend you, if you are practicing for this July, you can take a 3-month package. Yes, if you are aiming for your December 20 or you want to give it up with June 21, definitely I wish you to go with your 6 months or else a 12-month package. If you think that, yes Anuja, if you think that you are a very slow learner and you are just an MSc first year, I highly recommend you to go for a 20 more month package because it is going to be very much cheap at 12,600. Yes, absolutely. Yes, Shrisha, I am taking your special classes as well. Yes, I am taking the special classes. So I will be taking it up on Monday. Yes, and Wednesday. Right? 9 p.m. I am taking up classes there. Right? All right. Definitely, everybody. So yes, and this is my code SaranyaLive. You can use it for the 10% off. All right, everybody. I'll catch you up in another interesting session. Bye bye. Take care till that. Yes. Yes, everyone.